So our next speaker is Shyam Prabhakar from Genome Institute of Singapore, and again he's one of the founding members of HCA Asia. And go to it, Shyam. Thank you. Welcome.
Thank you, Al. Excited to be at round 5, I think, of HCA Asia meetings. I think we've come a long way since Jay and Piero kicked this off in Okinawa 4 years ago. And I hope you'll all see that the consortium has been gaining momentum and starting to make real contributions to the community. So before talking about HCA Asia per se, I'm going to zoom out a bit and talk about the Human Cell Atlas Genetic Diversity Bionetwork, which Jay introduced. And thank you, Jay, also for introducing the HCA Asia, the overall goals and so on. It really makes my life easier. And I knew you would do that, so I cut my slides short. So very briefly, in the Human Cell Atlas Genetic Diversity Network, we recognize that current genomic atlases are predominantly European and Western, but human beings are diverse and global. So, and it's not just that we're diverse and global, that diversity and geography has clear consequences for disease biology. Okay, so it's not merely a curiosity. So if we want to build a human cell atlas, we have to build an atlas of all humans. And that's really important if we want to translate the science that we're doing to populations all over the world rather than only a handful of populations, right? But I want to add to that also that diversity is a scientific frontier. You know, every, I would say, setback is an opportunity, right? The fact that no one looked at this before very seriously in a scientific context means that we have a chance to do something new and something exciting, right? And I think that's where HCA Asia can make a very powerful contribution. We're very well placed for that. So Asia has 60% of the world's population. The 2 most populous countries, tremendous diversity along all axes, right? Genetic diversity, ethnic, cultural, environmental, socioeconomic, you name it. And so Asia really has a big role to play in the human cell atlas, and particularly in the GDN. And that was really the impetus for starting Human Cell Atlas Asia. And very happy to describe our first pilot project, or flagship project across Human Cell Atlas Asia. And the impetus for this was that, okay, we all agree about the broad goals of Human Cell Atlas Asia. How do we put them into practice? And putting them into practice means many things. It means training. It means community building, networking, and so on, capacity building, and so on. But perhaps at the heart of all of it, what this network means, or what HCA Asia means, is we have to do something together, not just as individual PIs or as individual institutions, but some kind of science and some kind of output that is a collaborative effort, ideally across countries rather than just one single country. And that was the impetus for AIDA, the Asian Immune Diversity Atlas. And the goal of AIDA is to produce the world's first single-cell atlas of Asian immune cell states using RNA profiling, attack, genotyping, BCR profiling, TCR profiling, so on. Initially starting with blood, because that's the easiest to homogenize across and standardize. And I'll say a lot more about that, because that's a real challenge in these transnational consortia and collaborations. So AIDA has already racked up 9 countries actively participating in this effort. Singapore, Japan, South Korea, the initial 3, and then India and Thailand join, and then Russia, Sri Lanka, Pakistan, Saudi Arabia join. And it's an open consortium, so we welcome everyone. And we'd really like to cover as much of the diversity as we have in Asia as possible. And we want to look at the effects of ancestry, age, sex, environment, lifestyle in AIDA. But starting first with a healthy baseline. Okay, so AIDA will be profiling samples from healthy or nominally healthy individuals and using that as a baseline for disease omics and precision medicine in Asia. And as you can imagine, everything from autoimmune disease to infectious disease to cancer, metabolic disease, almost everything has an immune dimension, right? And you need a healthy baseline for that. And I'll show you some initial results, which I hope will encourage you to, that there is a tremendous amount of diversity. And so we really need a diverse set of healthy baselines, right? Just like ultimately we need many reference human genomes for different parts of the world and different ancestries. And then the initial thing we're doing with this healthy baseline in AIDA is we're identifying biomarkers and inflammatory mechanisms in metabolic disease across multiple countries. So that's the grand scope. I can assure you it was exciting and also ambitious and challenging to do this across so many countries. Tremendous organizational and logistical effort and technical. So I'm going to get into some of the technical aspects of this so that maybe other people who are coming along who are thinking of launching their own HCA Asia consortium or plugging into AIDA can get a sense of what we've been through and some of the challenges we face and how we address them. Okay. So multi-site, multi-country single cell study is very challenging, and the elephant in the room is batch effects. Okay, if different people in different labs all over Asia generate the same data, right? Is it really the same data, or do you have batch effects from country to country, just from technical differences? And so we want to address that or mitigate that as much as we can. And I'll say right at the outset, we cannot eliminate lab-specific batch effects. I think it's just not possible, right? But we can make it smaller and smaller and smaller, and then maybe bioinformatically address it. So standardized sample collection, standardized sequencing protocols across the sites, standardize the questionnaire, because the metadata are as important as the data, right? You want to correlate the molecular data to the metadata about the donors. And then another not unique, but unusual thing we did in AIDA is all the countries and all the labs are processing the same set of reference samples. So that if you have major technical biases in your single cell processing, you'll catch that, right? So at least it's a diagnostic for how well our harmonization has worked. And then this is all federated, okay? Everything in blue up here is federated, meaning each country does it in their own place. And that's an important part of our philosophy to encourage and support local research, right? Rather than centralizing everything. But then we don't feel there's a huge, cost benefit loss to centralizing DNA sequencing, more and more commodity now. And that takes one aspect of technical variation out of the picture. And we also centralize the low-level primary data analysis. So there's no huge benefit to having the low-level data analysis be done individually in all parts of the world. In fact, that can create a lot of disparity, right? But then the secondary data analysis is federated, and each institution in each country can take the data and analyze it for their own national prerogatives and their own scientific interests. Of course, another major challenge is that data sharing is already a challenge. Okay, if you want to ship data to the Human Cell Atlas (TCP), you have to make sure your ethics and consent forms allow it. You have to make sure your local laws allow it. And really, these are different in different parts of the world. These are different. Ethics are different from institution to institution. Laws are different from country to country. And so in a large transnational consortium, these are things we really grapple with. So, but I'm glad to say that within AIDA, we've achieved some kind of understanding of how to make this consortium work and how to contribute data despite the disparity in these areas across countries. But I'm not saying we've nailed it. There are still some countries, and I think some of the countries I might be referring to, where you cannot export your data, or you almost cannot export your data, the raw data, to another location. And so that is really a challenge, right? And I think that HCA Asia has to come to grips with that. HCA has to come to grips with that. And of course, communication, lots of meetings, lots of emails. But I assure you, the fun part of it is that you get to know other researchers in Asia. And the best way to get to know people is to collaborate with them. And it really strengthens our science. So part of harmonization is we share protocols not just within IDA, but on protocols.io, so that anyone, anywhere in the world can download our methods and use them. And hopefully, even in other parts of the world, there'll be harmonized data generation, and we can compare to their data sets. Another thing we do to reduce batch effects and also scale up, okay, make the assay cheaper and faster, is Jimmy's MUX-seq, as he calls it, method of pooling cells. So here each color is a vial of blood or PBMCs from one donor. You thaw those PBMCs, you count them, and then you pool them into one single tube. And then you do just, say, 2 10x genomics reactions to generate your single cell libraries, and you sequence it. And then you bioinformatically demultiplex that. So you say, okay, if this cell has lots of reads with genotypes from the matching genotypes of the red individual, okay, then it's a red cell. If the reads match the genotypes of the light blue individual, then the cell came from the light blue individual, and so on. So that's quite nice and makes it much easier to scale up. I highly recommend this method. And so this initial data set I'll talk about for the rest of this talk was generated across 3 countries. So Singapore, where I work, as well as Japan, so driven by Jae-shin at RIKEN, who just spoke, and South Korea, led by Woong-Yang Park at the Samsung Genome Institute. So all 3 countries use this method and the same harmonized protocols to generate PBMC single-cell RNA-seq data and some single-cell attack-seq data as well. And then we did some fairly systematic data analysis on that. So it's a multi-stage data analytics pipeline. It's not just one simple conventional Seurat workflow. I won't go through all the details, except I'll say the red box and the blue box are your 2 different stages. Okay, so you have 2-stage QC and 2-stage clustering. In our first stage, we do kind of generous QC and take any half-decent cell as a cell, and we remove doublets, and then we identify the major cell types using reference-based clustering. That's our RCA algorithm. But we're not done. Okay, so then we do a second round of QC. For each major cell type, we do even stricter QC on number of detected genes and % mitochondrial reads with cell type specific cutoffs. And then we visualize that, and we use Seurat on each individual cell type to do integrative analysis. And we find that the second round of QC really dramatically improves the quality of our downstream analysis. So here's a uniformity we were very relieved to see. So each box in this box plot is one batch of one pool of 16 samples in MUX-seq. And yellow is Singapore, red is Japan, blue is South Korea. You can see they're all hovering around per sample, per reaction, around 1,000 cells. And we did 2 reactions per sample. So that's 2,000 cells per individual, which is what we wanted to achieve. We're glad to say we achieved it. Some outlier individuals and batches, which we just discarded. Right? And then this is a cell type specific QC I told you about. So T cells, B cells, monocytes, dendritic cells, all have their own different distinct QC cutoffs. So the black dots are the cells that are within your QC cutoffs, and the light gray dots are the cells that you discarded. And you can see that the cutoffs are different for different cell types. And that's really important for your, in the second round, QC is really important for data quality. So at the end of all of this, strict filtering and so on, what do we have? We have 1 million cells from almost 550 PBMC samples, single-cell RNA-seq, across 3 countries. What does it look like? So here you go. This is your 1 million cell UMAP. T cells, myeloid cells, including CDCs, dendritic cells, B cells, good bit of platelets, plasmacytoid dendritic cells. And we were very pleased to see that the naive CD8 T cells separated out. That's an indicator, you can say, of the quality of your data and the quality of your data analysis. Very pleased to see also that the T cells had a lot of structure. Okay, this T cell UMAP doesn't look like a sausage. So a lot of structure means that you are able to resolve the subtypes much more easily, right? Much more accurately. And you can see here that the cells that we designate as T cells are really T cells. They express a T cell receptor. Cells that we designate as B cells are really B cells. They accept the B cell receptor. Even the plasmablasts express the B cell receptor. So that's great. And we did BCR and TCR sequencing on all these samples, in addition to 5' RNA-seq. And Jay mentioned the advantages of 5' single-cell RNA-seq. The major advantage is that you get to see where the 5' end of the transcript lies, in most cases. And you get the 5' RNAs that tell you the locations of some enhancers, your transcribed enhancers. And so you get more bang for the buck. You get the transcriptome as well as the cis regulatory element activity in the same cell. And so at Jay's suggestion, actually, very consciously followed this 5' single-cell RNA-seq strategy within IDA and hope to reap the dividends with Chung's data analysis pipeline. So what do Asians look like? So this plot I showed you is across all 3 countries, right? And in these 3 countries, you have 5 ancestries, at least. So Japanese, Korean, and in Singapore, you have Chinese, Malay, and Indian ancestry. It's quite a multi-ethnic country. So 5 ancestries at least. And so that's what we have here. How many B cells? What's the proportion of B cells, monocytes, NK cells, plasmablasts, and so on in each ancestry? Now, the first thing you see, and we use these Caucasian samples or European origin samples as controls, okay? And we profile the same controls in all 3 countries. So the first thing you see is that the European samples are really the big outlier. Okay? So they have way more B cells, more monocytes, substantially fewer NK cells, and it makes sense. Genetically, Europeans are most distant from Asians. And so even in terms of their blood cell counts, it makes sense that they're most distant. But I would say we cannot completely rule out that this is simply because the European samples were collected in one way by a private company, whereas the Asian samples were all collected in a different way within IDA, right? That's quite possible. It's hard to disentangle biological difference with the technical difference. But within Asia, you can say Chinese and Indian ancestry samples were collected side by side in Singapore. There's not much likelihood of a batch effect here. And yeah, I should say these samples were Chinese, Malay, and Indian samples were all collected by my collaborator, John Chambers, and his lab at LKC Medicine in Singapore, exactly side by side. So you can see here that Chinese ancestry individuals seem to have more NK cells than Indian ancestry individuals, and it's not massively statistically significant, but it is significant. Then Korean individuals seem to have a substantially higher proportion of plasmacytoid dendritic cells. This again, we need to validate. It could be because there's some unforeseen and unknown, unique way in which blood samples are collected in South Korea. We cannot rule that out. We have to look at our controls and check that. But this is an example. So I should say everything I'm showing you here is very preliminary data analysis, but it's an example of the kinds of science you can do when you do this kind of data generation. Okay? So major cell type proportions, they are not the same. They're more or less the same, but they're not exactly the same across countries. Right? And that's something very basic, right? You do your CBC analysis in your blood test, and I'm fairly confident that the baseline that people use across Asia is more or less the same, right? But maybe it should be changed and should be country specific. Then what about age? Does age have an effect on these cell type proportions? We found out not much, okay, at least in this age range of around 20 years to around 70 years. Not much of a correlation between age and cell type proportion, except plasmacytoid dendritic cells, intriguingly, there's a negative correlation. They go down with age. And this is quite statistically significant. We'd like to see now, is it across all Japan, South Korea, and Singapore, all 5 ancestries? Is it same in male and female? And things like that. So those are the second order analyses you can do here. But already we're seeing some biology here. Speaking of male and female, that was a big, maybe not a surprise, but a big result from our initial analysis. The blue boxes are female samples, the red boxes are male samples. You're seeing really massive differences in cell type proportion between males and females, okay? So for example, Indian and Malay donors, females had substantially more B cells than corresponding male donors. You see the same effect in Chinese donors in Singapore, but it's not as big. And then a much smaller effect in Japan and South Korea, which is very, very intriguing for us. Okay, so South Korea has a slight increase in females versus males. But in Japan, and in Japan also there's an increase, but it's much smaller, right? So it seems like the sex-specific effect is also modulated by our ancestry, which is intriguing. Then if you look at T cells, you see again females from, say, Chinese and Korean females have significantly more T cells. Japanese females also have more T cells. But intriguingly, so there was no such effect among female Malay donors, right? There's a little bit in female Indian donors. So again, this could be modulated by sex as well as ancestry. It's a complicated interaction. Then you see monocytes. Indian females have fewer monocytes, but again, that's not shared. That's not shared. So Japanese females have more or less the same monocytes as males, but Chinese females have fewer monocytes and so on. Again, we're seeing this complexity of sex and ancestry interacting. NK cells, even more fascinating. 4 ancestries have fewer NK cells in females, but Malays have more NK cells in females. So it's really remarkable. So we hope to dig our teeth into this much more. But I would say it's really a playground for doing very basic science and seeing how ancestry affects us in a way, affects our cells. So now at a high resolution, this is just gross cell types, right? B cells, T cells, plasma B cells, so on. Now we want to look at higher resolution because we have single cell data, right? So here's our UMAP again. These are T cells, these are CD8 naive T cells, these are B cells, platelets, myeloid cells. Now, this is a more high resolution way of saying, let's say, are Indians different from Chinese, are Malays different from Chinese ancestry, all in Singapore, right? So what does this plot show? Orange means that you have an enrichment, okay? And so this color orange means that it's a 4-fold, 2^2, 4-fold enrichment. So Indians are 4-fold enriched in this type, this subtype of T cells, of naive T cells, CD4 T cells. And they're enriched in this subtype of B cells. But they are depleted relative to Chinese ancestry individuals in Singapore, but they're strongly depleted, 4-fold or more depleted in this subtype of T cells and here in NK cells, right? But only some portions of NK cells. But if I take all CD4 naive T cells, there's some blue and some orange. So Indians and Chinese won't be that different, right? But at a higher resolution, you can see that there are intriguing cell state differences between Indian and Chinese. Same between Malays and Chinese, right? There are differences. And then if we compare Indians and Malays also, they're different. And so that's really fascinating. So for example, Malays seem to have an enrichment of CD14 monocytes with this phenotype, with this gene expression phenotype over here. And so can we look even, so I don't think this has ever been described, okay, these fine-grained ancestry differences, and that's the power of single cell analysis, and that's what we hope to uncover in this consortium. Now let's look more broader. These are all 3 ancestries in Singapore. Let's compare to, and using again Singaporean Chinese as their reference, looking for fold changes. compare Korean donors to Chinese donors, or Chinese ancestry donors, Japanese donors versus Chinese ancestry donors. You can see the blues are more blue down below, and the oranges are more orange, which means that, you know, if you take it at face value, then South Koreans and Japanese are very, very different from, you know, Chinese origin Singaporeans, much more so than Indians, Chinese and Malay origin individuals in Singapore are, right? Because these are light blue and light orange. But we felt it unlikely, right? So in terms of ancestry, Koreans and Chinese ancestry are the closest. They should not have among the strongest differences, right? So we suspect that there's some amount, even though we really did our best, we want to provide this, you know, even though it not as something that makes it look bad, but just as a reality that you are going to have country-specific batch effects if you do this yourself, okay? And I think the field is wide open if you have some bioinformatic methods to address this, and the conventional batch correction methods won't address this, okay? We've already used Surat integrate data to generate this plot. It has not addressed the country-specific batch effects. And I can explain why in a separate conversation. So this is a major challenge for the field. But at least within one country, we can say that the ancestry-specific differences we find, we believe they're real and they're not affected by batch effects, right? So we want to harmonize data analysis as much as possible, batch correction methods, enhance RNAs, differences, all kinds of diversity, expression QTLs is a long road, exciting early results. Finally, I'd like to show off a new award we just received. I'm very pleased to announce that the AIDA Consortium has received one of the CZI, Ancestry Networks for the Human Cell Atlas grants. And this is to 8 countries represented by myself and John Chambers in Singapore, Jay Shin, Wong Young Park, Partha Majumdar, Varadom, Yulia, Anuradhani, Khadija in Thailand, Russia, Sri Lanka, Pakistan, respectively. 20 ancestrally diverse population groups from 8 countries. We'll be profiling 2,000 healthy individuals. And this, we feel, will really flesh out Asian diversity and, of course, welcome even more participants. So I hope you'll take this as an example of what Asia can do, or Asia-based researchers can do when they come together and work across boundaries. With that, I'd like to thank all the team members who contribute to this. Particularly, I want to point out Xiaoshuan Kuei Lin, who is a bioinformatician for Singapore AIDA, and Deepa Rajagopalan, who drove the wet lab part of AIDA in Singapore, and team members from my lab and from Recon SGI LKC as well. Thank you.
Okay, great. Thanks for that, Shyam. That was really enjoyable. We do have some questions starting to come through. And the first question is, how can you differentiate between the effects coming from ancestry versus the environment?
Fantastic question. And the only way we can do that is with a 2x2 study design, right? So you have same ancestry individuals from 2 environments and same environment individuals from 2 ancestries. And so at least in Singapore, we have multiple ancestries, same environment. That's wonderful. Then with the Ancestry Networks grant, we have Partha driving a component in India. So then we have Indian ancestry people in 2 different environments or 2 different geographies. One is in India, one is in Singapore. And actually in Sri Lanka also, the ancestry is not that different. For Tamils in Sri Lanka, very similar ancestry, right? So that's how, with this 2x2 study design, you can start to tease those things apart.
We got a question from Christine Wells. How will you account for population diversity within each of the countries in AIDA? China and India cover huge population groups, and Singapore is such a melting pot.
Great question. There's no question that China and India have way more diversity in more or less any form than Singapore, which is a country of 5.5 million people. This is a very long journey, and we're just starting down this road. So Partha's team is starting up. But I would say I said 8 countries, right? But I mentioned 20 population groups. So in each of the Ancestry Networks countries that are funded now, we are going to profile as much genetic ancestry diversity and geographical diversity as we can already in this group. But of course, it'll be very, very incomplete, and you just go from one step to the next, right? There's a long way to go for sure.
I had a question. So I mean, one of the major findings you presented was differences across the cohorts and differences between sexes based on single cell profiling. How much of that is already known from, say, flow cytometry, and have you generated full blood counts on the same patients?
Excellent question. So from facts, to be honest, this is very new. We got these results a few days ago, okay, at the end of last week. We now have to comb through the literature to see how much is known with facts and how much is not known. But certainly the interactions between sex and ancestry, particularly for Asian ancestries, I doubt it'll be there in published facts data, but we have to dig in. And I forgot your second question.
Oh, and whether
CBC.
you're generating
CBC.
count data yourself.
Yes. We are, and that is something that we can use to corroborate for sure.
Cool.
But we have not analyzed that data yet, and okay. I'm not sure we've uniformly generated or fully generated that data either. We have to dig that up now.
Okay, we are over time, but there's one last question from Engli Zhu. Are all the participants unrelated to each other?
In Singapore, it's quite likely that, well, in all countries, some of them will be related to each other. But we have experience from the National Precision Medicine Program, and in fact, our Singapore IDA cohort is from Singapore's National Precision Medicine Program, and that's a cohort that John Chambers is driving, who's speaking in this session very soon. And we've looked at genetic relatedness from the genome sequencing of those individuals. It's not terrible. So, and we can identify first degree and second degree relatives from the genome sequencing data. So I don't think that'll be a huge factor in our data analysis.
Okay, cool. Thank you very much, Shyam. And thanks both to Shyam and Jay for fantastic introductory session. And now we've got a 10-minute break. And please join us back promptly for that lesson session, which will be chaired by Shyam and Aki Minoda. And the first speaker of the session is Goji Go from China. Thank you.